This is my second time recording this. I meant to hit restart. So I'm going to post the link to this in the description, but I'm going to try to go over this as quickly as possible because it is important to know these terms for the second microbiology exam over chapters 4, 5, and 6. Maybe yours is over more chapters. Sorry, this is just how it's broken down for me. So we're going to start with listing all the structures the bacteria possesses, and this is starting in chapter 4. So you have the cytoplasmic membrane, which I may shorthand to cytomembrane, the cytoplasm, ribosomes, a cytoskeleton, and one or a few chromosomes. And then 4 structures that some but not all bacteria possess may be cell walls, cell membranes, cytoplasms, ribosomes, and the chromosomes. Remember, you have rough ERs and you have smooth ERs. And that plays into your ribosomes in your bacterias. Describe the 3 major shapes of bacteria. So I have a picture here, but for those who like to study with the speech on, you have bacillus, coccus, and spirillum typed out. And you can just see what they look like. Describe more unusual shapes of bacteria. So you can see 6 different kinds here. Vibrio is mentioned a lot. I would know what the Vibrio looks like. Basically, they say it's a long, broad, kind of bean-shaped. And Spirochetes is a good one as well. 4 terms to describe bacterial arrangements. So you see in the picture here, Coccus 1, Diplococci 2, Tetrad is 4. Sorry, trying to roll my R there. And then Sarcina is going to be 8, or I believe it goes up to 8 or 16. Streptococci and Staphylococci. Staphylococci is a pretty common arrangement. Describe the structure and function of 6 different types of bacterial external structures. So you have all of them listed in this graph here, but I wrote out flagella, axiofilaments, fimbriae, pili. I don't know why I didn't finish it. But you'll see nucleoid, plasmid, and a spore granule. You'll see the chemical composition and the function, which is great. Explain how a flagellum works in the presence of an attractant. So it'll tumble, it'll run, it'll tumble, and it'll run in the direction of the attractant. Obviously, attractant will want to go to it. But this picture is a good example. Differentiate between the 2 main types of bacterial envelope structure. So you'll see there's a thick purple on the left, which is gram-positive, and then there's not so much purple on the right, which is gram-negative. So gram-positive, the big difference is going to be the thicker wall, and as a cytoplasmic membrane, and then you have the gram-negative has 3 layers, cytomembrane, thin wall, and then it has an outer membrane as well. And you can kind of look at that there. That's straight from the book. Discuss why gram-positive cell walls are stronger than negative. Obviously, they're thicker. This is another example. They're less sensitive to lysis, which we'll talk about all the way at the end. But basically, the destroying by viruses. Name a substance in the envelope structure of some bacteria that can cause severe problems in humans. So that'll be polysaccharides and LPS. They're referred to as endotoxins. I can't really find anything on endotoxins here, so here you go. Fun picture. Identify 5 structures that may be contained in bacterial cytoplasm. Again, just a fun picture. You can see the cytoplasms there. Nutrients, chromatin body, ribosomes, granules, and fibers. And then detail the causes and mechanisms of sporulation and germination. So sporulation, think ulation, formulation, spore forming. So endospore formation starts with the depletion of nutrients, and I have this whole picture here, which is so fun. So then the chromosome is duplicated and separated. The cell is separated. into a sporangium and a forespore instead of a septum. And so that's important to know. And the way it, like, you can kind of think of it, it's like it's separating into like two weird shaped boobs. And I'm like, damn. And then sporangium, like, I don't know. That's just how I do it, which is probably the stupidest thing in the world. But if you are desperate for some way to remember it, so it's separated into the two, forespore, sporangium, and then it engulfs the forespore, and then it begins to synthesize, and you'll see your early endospore by step 5. And then the cortex and outer coat layers are deposited. You'll have a mature endospore by 7, so forth. List some differences between archaea and bacteria. So you can see here a few different things. Flagella of archaea are solid, bacteria is hollow. They don't have archaea does not have a peptoglycan wall, bacteria does. There are no introns present in bacteria, but there are in archaea. No ribosomal protein in bacteria versus is in archaea. And then knowing the difference between Berge's Manual of Systemic Bacteriology and Determinative Bacteriology is important. And here you'll just see some classification bases off the systemic bacteriology. But so systemic bacteriology presents bacteria and archaea relatedness, combining with phenotypic info with RNA sequencing to classify them, versus the other one is based entirely off of phenotypic characteristics. So it's important to know what a phenotype is. Name 4 divisions ending in cutes and describe their characteristics. This isn't necessarily very important to the exam, but it is good to know. So granulocytes, firmicutes, tenoracutes, mendicutes. Define a species in terms of bacteria. This is just a fun picture. Remember, species is the most specific you can get in bacteria. So a collection of bacterial cells, all of which have an overall similar pattern of traits. Microbiologists use subspecies, strain, or type to designate bacteria with the same species with differencing characteristics. And then you need to know, like, in that relation, like, how specific is a strain. Strains cannot be different from the original in any way, shape, or form. And then relate bacterial, archaeal, and eukaryotic cells to the last common ancestor. You can see here it's likely a large organism with loose boundaries and has RNA as genetic material. List the types of eukaryotic microorganisms and denote which are unicellular and which are multi. This is a fun picture that doesn't blow up well, but it'll show you all the answers there. Differentiate between cilia and flagella in eukaryotes, and differentiate flagellar structure among bacteria, archaea, and eukaryotes. So this shows you cilia and flagella structure. Eukaryotic flagella is thicker, structurally more complex, and covered by an extension of the cellular membrane. Cilia is shorter and more numerous. Cilia are only found in one group of protozoa and certain animal cells. Describe the important characteristics of a glycocalyx in eukaryotes. So here you'll see the glycocalyx capsule, and basically there's a capsule, there is a slime layer, and that's your fun slime layer that you're going to want to remember. It's an extracellular matrix network of fibers, a slime layer, or a capsule. List which eukaryotic microorganisms have a cell wall, fungi, and algae. And there's a fun fact for you. And list in similarities and differences between eukaryotic and bacterial cytoplasmic membranes. So here are some distinction characteristics between eukaryotic, prokaryotic, and viruses. But in addition to the types of bilayer of phospholipids and sheet proteins are embedded, eukaryotic cytoplasmic membranes contain sterols. Cytoplasmic membranes of eukaryotes have the same function as those of bacteria, serving as a selectively permeable barrier. Describe the main structural components of a nucleus. Here you'll see the nucleus. You'll see the pores, the envelope, the nucleolus, and the endoplasmic reticulum. And it's separated from the cell cytoplasm by the nuclear envelope. And then inside the nuclear envelope is a substance, the nucleoplasm. But I don't think that's super important to know. The plasm part, sorry. Diagram how the nucleus, the endoplasmic reticulum, and Golgi apparatus all work together. So here you'll see the transport process. It starts in the nucleus, and it goes in instructions in the form of DNA and proteins, right? So then it's going to go to the endoplasmic reticulum, which you'll see here. This is the RER. And it's going to synthesize and transport to the Golgi, which looks like the pancakes, and it'll get sent to the final destination. The Golgi does often modify by polysaccharides and lipids, but in the end, it pinches off finished condensing vesicles, which you can see here, and there's secretion by exocytosis. Explain the function of the mitochondrion. Obviously, you know the mitochondria is powerhouse of the cell. Why power, energy relates. You can see how the mitochondria is, has the inner membrane, outer membrane, cristae matrix. It holds enzymes and electron carrier of the aerobic respiration. So when you think aerobic respiration, think about the mitochondria. Don't get it confused with anaerobic. Discuss the function of chloroplasts. So here's a picture of chloroplast. It's a double membrane envelope. It has 70S ribosomes, soma, thylakoids, granum, DNA strands, important to know thylakoids and granum. And I just don't have time to read all this, or YouTube will hate me, but explain the importance of ribosomes and differentiate between the types. So here you can see in the picture, it's composed of rRNA and proteins scattered in the cytoplasm or associated with RER, larger than prokaryotic ribosomes, and function in protein synthesis. List and describe the three main fibers of the cytoskeleton. So here you see the picture of the cytoskeleton as the actin filaments, the intermediate filaments, and the microtubule. So the actin filaments are long, thin protein strands. The intermediate are rose-like structures. Really, I think they're more like those kids' cups that you're not supposed to be able to knock over. But we'll go with rose. And then microtubules are long, hollow cells that maintain the shape of eukaryotic cells when they don't have cell walls. Very important. And there's three general features of fungal anatomy. So you have the hyphae, the pseudohyphae, and dimorphic. Dimorphic's important to know. And then hyphae, it's long thread-like cells found in the bodies of filamentous fungi or molds, and that does come into play later, which you'll see. So important to know. And then differentiate among the terms heterotroph, saprobe, and parasite. I remember this by heterotroph fungi acquire nutrients from a variety of organic materials called substrates. I think of like using a troph, if I'm thinking of the right thing, like out there farming organic materials. And then probe, you probe the dead. That's my reasoning for that. So they get their nutrients from the dead plants. And then parasites, pretty straightforward. They live on the bodies of living plants. Shouldn't really need anything for that. Explain the relationship between fungal hyphae and the production of a mycelium. So mycelium, like I said before about mold, the hyphae makes up the mycelium. It's a colony basically. So you'll see here, this is the mycelium, and yeah, it's fungi growing in loose association or in colonies. Describe two ways in which fungal spores arise. So you have asexual spore formation, and then you have sexual spore formation. There are pictures of asexual spore formations here, but it's mitosis or meiosis here, basically. You need to know the difference between the two of those. List two detrimental and two beneficial activities of fungi. So basically it can kill plants, corn, grain, rot produce, cause human disease, but it also puts organic matter back in the soil, helps plants, creates alcohol, yeast, and can flavor some foods. And then protozoan characteristics that illustrate why they are just thrown generally around. They lack true tissues, so they're unicellular or colonial and you can see they just kind of group together. Here are some specifics: modes of contaminations, life cycle symptoms. Just kind of interesting. List 3 means of locomotion exhibited by protozoa. So locomotion is moving, obviously. Pseudopods, you need to know that means false feet. This is what they are: blunt, branched, long, or pointed. Sorry, I keep thinking I'm scrolling down. Flagella are attached to the long length of the cell by an extension of the cytoplasmic membrane, and then the cilia are distributed over the entire surface of the cell. Very important to know. Explain why a cyst stage may be useful in a protozoan. So here's the whole thing. It goes from active to early to mature. Then the cyst breaks open and it goes back to active. It's for protection until favorable environments come around. So basically, it forms a cyst in a hostile environment. Give an example of human disease caused by each 4 types. Not super important to know, but they're list the 2 major groups of helminths and provide examples representing each body type. So here are some different options pictured. The way I learned, so tapeworms, cestodes, flukes or trematodes, anything ending in odes, at least for the purpose of this chapter, is going to be some sort of worm and some sort of helminth. Summarize the stages of a typical lifestyle: fertilized egg, larvae, adult stages. That's all 3 easiest way. And then this is some information. Explain what it means when viruses are described as filterable. Basically, they can still be infectious after going through a bacterial filter because they're so small. And this is about sizes for reference. And then instead of alive or dead, you say active or inactive. Here's the process of an active virus. List the 5 properties of viruses and that distinguish them from living cells. So here you can see virus versus living cell, structure, reproduction, genetic material. So virus does not grow and develop, it does not obtain or use energy, it does not have a response to environment, it does not change over time. Whereas living cells will. Virus can have one or the other. DNA has DNA and RNA. Living cells have DNA and RNA. So that's good to know. And then discuss the size of viruses. Here's another picture showing size like the last one. They're much smaller. Describe the function of viral capsids. The shell surrounds the nucleic acid. That's the best thing for it. Each capsid is constructed for identical subunits called capsimers that are constructed for protein molecules. Distinguish between enveloped and naked viruses. So here you'll see non-enveloped and envelopes have spiked. And is composed of phospholipid bilayer proteins, which often play a role in host recognition, meaning what host can they attach to. And then they have a geometric capsid assembled around the nucleic acid strands for naked. Explain the importance of viral surface proteins or spikes. It's used in the process of absorption, essential for the attachment of viruses to the next host cell, target for antibodies, very important. Compare and contrast the composition of a viral genome to that of a cellular organism's genome. The number of viral genes is small compared to that of a cell, and they possess only the genes necessary to invade host cells and redirect their activity. And then it says, diagram the possible configurations nucleic acid viruses may possess. Couldn't really do that, but you'll see here I used plus for positive and then minus for negative. And demonstrate how family and genus names in viruses are written. Italics, you'll see here family, italics. And so like this is what the term is for Ebola. What the heck? Ebola. Stop while I'm ahead. And then the 6-step life cycle of animal viruses is very important. So I have it written out, and then you can also see here in the diagram. You have adsorption, penetration, uncoating, so it frees the viral DNA, Synthesis, assembly, and release. I'm sure there's a mnemonic for that. I have a terrible one that I cannot say on YouTube. Define the term cytopathic effect and provide one example. So here you'll see a whole picture of it, which is very interesting. My favorite is poliovirus because look, there's nothing left. Not the polio is good. Virus-induced damage to the cell that alters its microscopic appearance. Provide examples of persistent and transforming infections. Persistent is viral infections maintaining a carrier relationships, so it's not going to lyse for up to a few weeks or the whole lifetime. As you recall, lysing will kill the cell. Transforming, the virus enters the host cell and permanently alters its genetic material. So it kind of refers to oncogenes, oncoviruses. So those lead to cancer. What's the difference between lytic and lysogenic? What are both these? They're both methods of viral replication. It is important to know lysogenic and lysogenic conversion. And then what temperate phage DNA is. And so basically, lytic bacteriophage injects into the host with nucleic acid. The host stops the DNA replication and forces it to work for the virus. And then it ultimately lyses because of the enzymes released from the phages. And then lysogenic, they're not going to do the viral phages. They're going to stay in a temperate phage, which means it's basically inactive prophage. And it's lysogenic conversion is where the bacterium gets a new trait from the cell. And then those three principal purposes of cultivating viruses is important because you need to isolate and identify viruses in clinical specimens, prepare viruses for vaccines, do detailed research. Very important. Describe 3 ways in which they're cultivated: bird embryos, so in vitro, using live animal inoculation and cell tissue. Describe 3 non-cellular infectious agents beside viruses. So here you'll see viruses, viroids, prions, but you also have satellite viruses, which depend on other viruses for replication. And then what are prions? They basically become misfolded versions of normal brain protein, causing brain damage. So this is a diseased prion, and it's it went from a sheet to a beta helix. And that covers everything. I hope you can look over these 57 slides and do well on your exam covering 4 through 6. Or this is a good refresher. I did talk really fast. Feel free to go into settings and slow me down to 0.5, and that'll make it a lot easier to understand, but I was also kind of crunched for time. But yeah, thank you.